Morning everyone, I'm welcome you all to this day 15th I'm welcome you all to this day 15th CSIR Life Science question and answer discussion session. Today we are going to discuss about the Unit 5 Part C questions, that is Developmental Biology unit, which is very important unit in CSIR examination. Okay, as per our discussion, earlier we discussed the Unit 4, 5, 13, right? 4, 5, 13 nearly carries the 36 questions, the 36 marks. Okay, each unit carries 36 mark in a CSIR exam question. So we want to well versed in this Unit 4, 3 and Unit 4, 5 and 13. Once again I'm repeat, unit 4, 5 and 13 is very important units. Each unit carries 36 mark in the ACSR examination. In that, 28 mark is all about the part C questions. There are 7 questions are there in a question paper. They are asked 7 questions, nearly 6 to 7 questions in a CSIR exam question paper. So we want to well versed in that particular topic. And one more thing about this unit 5. In this unit 5 is related to it 4. Okay, you can continually study the both unit whenever you are start studying for CSIR examination. First you want to study the unit 4 and then continuously studying the unit 5. That will helpful to you. And one more thing, this unit questions from this unit also have a relevancy with the for unit 2. Okay, each many of the topics are covered with cellular organization is the unit 2. That is also relevant to this unit 5. So if you are studied in if you are studying the unit 5, that some portions are covered the unit 2 part, unit 2 questions, so that is helpful to you for the understanding. So please study the unit 4 and 5 is in very thorough manner. Next we will enter to the question. In this question you can see this is a basic question. In this, formation of digits and sculpting the tetrapod limb requires death of specific cells in a limb in a programmed manner. Okay, this is the formation of digits and the sculpting the tetrapod's limb requires the death of specific cells in a limbs in programmed manner. Which one of the following interaction could explain proper limb formation? That is the if this is the statement, we want to know the correct process. Okay, this process is very important that requires the death of specific cell. So final stage is apoptosis. Death is relevant to the apoptosis. So the final stage should be related to our the death, cell death. The final stage is become it's a death stage, and it should be in a programmed manner. That programmed manner, that is the proper staging. This proper staging is explained in the option A. First the stage is that BMB protein is involved in the cartilage process. Okay, this cartilage process, and then after the cartilage, the Wnt and beta catenin activates in the mode. After the activation, DKK-1 is activated by the catenin, beta catenin, Wnt signaling, and BMP and FGF. This all those things are acting the DKK1 pathway. After this DKK1 activation, that DKK1 activates the apoptosis process. This is a sequential programmed manner. Okay, it's a process. All this here they are confused with the other options. That is not correct in the case of the case of the specific cell in a limb in a programmed that should be death of the specific cell is should be in a programmed manner. That programmed manner is here. That is the BMP that activating the cartilage. That cartilage are activating the Wnt pathway. This Wnt pathway is very very important in the human cell system. In this human cell system, or this Wnt pathway, they need a signal molecule is achieved from the metabolism of the cell glycolysis pathway. After the glycolysis pathway, that receiving the signal and they are activating this cartilage. That is the one method. This cartilage is not it's in active mode, but due to this activation, that pathway, the apoptosis pathway, is triggered with the BMP and FGF. This BMP is it's a like neuronal stem cell. Okay, it's a group of the neuronal stem cells. It's helpful for the their production, stem cell productions. This BMP, whenever you are studying the BMP, it's very very important keyword. This BMP is specific. It's having the different group. In this different group, BMP4 is playing a vital role in a smart dependent apoptosis cell death. The very very important thing, it's a latest research concept. BMP is BMP4 induce the smart SMAD, the smart dependent apoptosis cell death. Okay, this is the very important concept in the latest research. This is an old question. Nowadays they may ask this kind of questions in your CSIR examination. So that is very important one. And another here they went the cart this the BMP, it's activating the process of this is the cartilage. Okay, in this cartilage process, it's dependent it's a normal natural cells. Okay, or natural cells. Because of this cartilent, okay, cardinal cells, that cells are activating the articular cartilage process. The loss of the articular cartilage, that makes the apoptosis. This normal apoptosis, okay, that activation of the Wnt pathway, that activating the cartilage, is promotes the normal apoptosis process. Normal apoptosis process means that is in a normal human cell, not in a cancer cell and any other things. This apoptosis of chondrocytes, okay, the chondrocyte, that is the chondrocytes are the major component in the cartilage process. That chondrocytes are make a death. Okay, this death stimulating the apoptosis process. That is sequential process. Wnt activating the cartilage. In this cartilage, the chondrocytes are there. The chondrocytes death is triggering the apoptosis process. That is a sequential process. This mechanism, this cartilage area, okay, this cartilage area, it's totally having the apoptosis process. But there are the enzyme called a caspase enzyme. The count of the caspase enzyme is in a higher number. This caspase promotes the apoptosis process. Okay, it's a very important thing in this apoptosis concept. And DKK1, and it's also the another important thing. This it promotes the it's specific for that. These are all the concepts: BMP, cartilage, Wnt. These are all the responsible for normal cell apoptosis process. That is this is process. Here they mention this lymph record. Okay, lymph tetrapod limbs are required. The lymph cells are specifically undergone apoptosis through the promotion of the this DKK1 cells. Okay, Dicops F. Okay, this one is a this DKK1 cells. In this DKK1 cells, it's make a aggregation of the proteins, and it's trigger the it's that complex formation, that caspase 3, caspase 8 complexes are promoted by this DKK1 signaling. The expression of this particular DKK1, it's also contribute to the cartilage process. In this cartilage process also known, we are all studied that promotes the upper concept about this pathway. So whenever you are studying the apoptosis, we want to understand this concept. This DKK1 is specific for, it promotes the apoptosis in. So this is the one important, but the sequential concept is very important, is all about the programmed manner. Okay, next all about okay, here it's very easiest question. If you are known the fundamental process, you can easily answer this question. In this question, first things all about the developmental steps. Okay, invaginations, invoculations, ingressions, delaminations, epiboli. Here these are all the terms. Here we having the what is that, and here we having the where it happened. Okay, this is the process, what is the process, where it has happened. These are all the combination. We want to select correct one. The first thing is the A. The C answer is option is a correct one. A is invagination. Okay, invagination is the inward movement of expanding outer layer, so that it spread over the internal surface of remaining external cells. Okay, here the concept inward movement of expanding outer layer, that it spreads over the internal surface of remaining external cells. In this invagination, we can find it in, that is the option called the this option. Okay, C. For C, that correct option is the this third one. Sorry, fifth one. Another option is they are providing the C. That is for C. Here the B value, invoculation. This invoculation, it's immigration of individual cells from surface to interior of the embryo. This happen. This immigration, that is the invoculation. Another option is a C. That means C column is here the mesoterms. A column, this A column 3 ingressions, and the B column is immigration of individual cells from surface to interior embryo. Then B cells is are mesoterm in a C origins. Okay, sorry, once again I'm repeat the correct answer. The correct combination is column A. This column A, third ingressions means that is immigration of individual cells from surface into the interior of embryo, and then C column, that is happen in the mesoterm of the C genes. This is a very important this kind of questions we can expect in the CSIR question. Okay, where it is happening in this common developmental biology mechanism, this process happen in the each and every explaining the each and every words. Invagination, this is the process which happen in the developmental process in the gastrulation. Okay, the gastrulation process, it's egg formation. This invagination, it's the production of tubes and local impulsings of the surface. Okay, this is the just a local infusion, that local infusions. Okay, that gap is formed in the gastrulation process. This process of invagination, helps to invaginate some organs in the cells, developing cells, that very important. This gastrula, formation of gastrula, by the infolding of the particular wall, or infolding, this infolding make nerves tubes in these cells. This particular invading folding of the particular part, this process called as the invagination. In this gastrulation, invagination is a very important step. In this process, that uh, we can visualize the morphogenetic process. It's helpful to understand the these are all about the this invagination process. And then invoculation is another step which involved in the this what called developmental biology. In this invoculation, it's a shrinkage of a particular organs. In this invoculation, its act is the infolding. Okay, invagination is the infolding. Here this invoculation is the enfolding, enfolding. Okay, it's in this process, that a uterus is transformed from pregnant to non-pregnant stage. This is the process called invoculation. In this process, the uterus is transformed from pregnant to non-pregnant state. It's this occurring after the parturitions. Okay, parturitions. In this process, the hypertrophy of the uterus has to be undone in the process. That is not happen the pregnancy process. That is the unfolding stage. There is no gastrulation. There is no that oocyte zygotes formations. In this process, its major role playing system is the RU. And next thing is the ingression. In this example also they mention the ingression. Ingression is in this process, it's normal process which happen in this developmental biology. Thus in this process, many changes in the location. Okay, that change the location of each and every location. Okay, it's one of the many changes in the location of the cell. So here they mention the immigration means it's a changing location, it's a moving immigration of individual cells from surface to surface into interior of embryo. That is the process. It's happen in the mesoderm in the CEO gene. And next concept called the delamination. In this delamination, we have the it's a mode of the failure. That is it's normal mode. In this biological mode, the separation into layers. Okay, it's a initial splitting of cells in the embryo. Its process called delamination. This is the initial splitting of cells into embryo. This delamination, it's a process which happen in the cell. It's in the embryo stage to the cell's developmental stage. It's a blastoderm splits into two pair of cells. Embryo blastoderm is splits into two layer of cells. This is the process. In this is the process which happen in this development of cells. Okay, next thing is epibody, and this epibody is another important thing which happen in the cell. It's having this here the epipoly and empoly. There are two things are there. Epipoly, that is here the epipoly, this is a EPI. This one of the cell movement that occur in the earlier embryo during the gastrulation. Okay, it's one of the process happen in the gastrulation, cell movement. Empoly, empoly is the process of the cells occur in the invagination of blastula to form the gastrula. The process, this is the process. Empoly is a process in which the cells are having they are formed into invagination of blastula to form the gastrula. This is the process. Epipoly is one of the cell movement occur in the earliest stage of the gastrulation. This is the earliest stage of gastrulation. This empoly is the blastula to gastrula formation. This main purpose of this gastrulation, we all know, is the position that 3 embryo germ layers are there. That is the endoterm, ectoterm and mesoterm. In this gastrulation, having the endoterm, ectoterm and mesoterm, these layers are later developed into the body systems. This ectoterm developed into the skin, nail and epithelium cells, and this makes a nerve systems. Likewise, our endoterms are developing into the our various parts, organs. In this specific processes, okay, ectoterm, endoterm and the mesoterm. In some of the this process, okay, it is having carries the various steps. Here the mention as some of the cell layers are the mesoterm. Some of the animals are formed between the endoterm and the ectoterm. That depends on the cell layers. Okay, next question. It's very easiest one. In this second question, this is the third question. Which of the following signal process, which one is not involved in the cellular movement and cytoskeletal change? Very easiest method. Here, this is they ask the question, not involved in the cellular movement. A very important thing, cellular movement. This is kind of cellular movement. This is one cell. These are all the another kind of cell. That cell move towards the membrane. B question, as per the rho A, okay, RHOA, this is the compound which involved in the cellular movement. Okay, so this is the keyword. This is the keyword called RHOA. Okay, next concept is the catenin concept. This catenin is involved in the it's passing the signals and allowing the cells from external cells from internal cells. So this is the concept which helpful for the cytoskeletal changes. And here next is the RAS-RAF pathway. This is not relevant to the our cellular movement. This involved in the cellular process. So this is the odd man out. This is the wrong answer. This is the not involved in the movement and cytoskeletal protein. So if you are facing this kind of question, we want to understand the concepts. Remember the concepts. Okay, this is the one concept. Here the cell is trying to move out. Here this rho RHOA is the another important in the cell migration, the cell adhesions. Here then catenin is involved in the process of cell signaling. In the cell signal is helpful for the cytoskeletal cell. But this gap, RAS, RAF, all those things are involved in the apoptosis pathway. That is a cellular process. The cellular process, it's odd man out. So this is the answer here. Okay, next one is, which of the following about the development of sea urchin embryo? It's a true. Okay, this they provided the four statement here. They asked the which one is the very important. Why they are this in this question itself? You can see many of the studies are on the this sea urchins. So this sea urchins having many medicinal effects that their systems are easily studyable. But these sea urchins are somewhat they are dangerous and harmful to the human, but so their processed materials are helpful for for medicinal purpose. In the latest few past decade, last 10 years, many of the researchers are focusing on the sea urchins. So you can expect this kind of questions in your exam points in the exam. So here in this sea urchin, in the case that embryo is embryo concept is here. We are studying the all about this developmental biology. So this they are studying this the sea urchin development. In this sea urchin development, these sea urchins are undergone into the 10 different steps. Okay, here first I'm reading the given answer. First one is the each blastoterms of 4 cell stage possess the position of the original animal vegetal axis. It's having the original animal vegetal axis, and if isolated and allowed to develop will form a complete but smaller sized larva. If it is isolated and allowed to develop means that will form a complete and smaller size larva. Here as I mentioned, here this is the one statement is a correct one about the sea urchins. The sea urchins are having the blastoterm of 4 cell stage possess the portion of the original animal vegetate axis. If we are isolated and allowed to develop means that forms a complete but smaller sized larva. Okay, so this is the statement about this sea urchin embryo. Before the session, before the study, we want to know the concept in the sea urchins, the cell development. In this sea urchin development, there are different stages are there. First stage is a 60 cell stage. That is the small of 4 arm larva that is developed into 8, that is adult rudiments. In this, after this stage of the development of this 4 arm larva that are producing the adult sea urchins, it's not having a scale, that is the biggest one. Before this formation of each and every crossing the 10 stages alone, these sea urchins are developed and they are developing into the full stage. This cell division form a single epithelial layer enveloping the blastoceole. It's followed by the production of formation of the gastrulation production. This gastrulation having 3 germ layers. Once again, repeat that single epithelial layer is enveloping the blastoceles, and the gastrulation producing the 3 germ layers in the sea urchin. And next thing about the next question. 2 cell embryo is made of blastomere A and B. If the 2 blastomeres are experimentally spreaded, all these cells to it would normally make. However, the B blastomeres is isolated. Isolation makes a only small fraction of the cells it would normally make. Based on the above observation, only which of the following conclusion is correct one? First is they are doing the a two-celled embryo is made into blastomere A and B. A two-cell embryo is developed into two blastomeres, that is blastomere A, blastomere B. This, if two blastomeres are experimentally separated, we are separated the blastomere A and blastomere B. The blastomere A is generated all the cells in a normal make, but but here the blastomere B is separately isolated and make a small fractions. Okay, they are make up small fractions from the B blastomere. That would normally, that this small fraction cells that not able to produce the larger one, that's make it's only producing the smaller fractions of cells in a normal make. What is the above the observation, what is the correct scenario? The A is the correct one. Conditionally specified means they are having the restrictions in their developmental system, that is genetically adapted, genetically modified environment. Okay, they are developing in a conditional manner. This conditional development, in this conditionally specified cells are having the different natures in a particular environment. Okay, this hot condition some cells can able to grow, and cold condition some cells are unable to grow. That is a conditionally specified. But this a blastomere A its cells are can grow in any environment, any conditions. Okay, next question. This it's very interesting concept in the developmental biology in the developmental biology. Here you can see the first question is development of vulva in C. elegans. Development of vulva in the C. elegans. It's initiated by induction of small number of cells by short range signals from the single induction cell. With referred to this, following statements are put forward. We are providing the PQRS statement. Which of the above statements are true? Okay, first we want to understand the development of vulvas. This development of vulvas is called from each and every process in the development of vulva. It's carried out in the C. elegans is initiated by the induction of small number of cells by short range of signals. This short range of signals are they are induced by the single induction cells. This single induction cells, okay, single induction cells means during the embryogenesis process the cells are collected individually and they are induced by particular chemical, or induced by the chemical processings. In this developmental process, the single induction, they having the they are facing the environmental change, they are facing the chemical change. This make a effect. This process, okay, they as per this process they are providing the statement. Here they provide statement P and S is right. P and S, here the P is when the anchor cells were ablated earlier in the development, no vulva formed. Second is a constitutive signals from the hypodermics heat inhibits the development of both primary and secondary fates, but it is overlaid by the initial signals from the anchor cells. This initial signaling process, we all know, this initial signaling process means that are developing the two cell stage. Okay, in that stage itself we can change we can make a change. After the embryo and castrulation develop, we can't change this step. So here they are changing the induction, single cell induction. It's in the initial signals from the anchoring cells. So this constitutive signals from the hyperthermic inhibition to the development of both primary and secondary fates, but it's overruled by the initial signal. So this overload make a single cell induction and the initial stage. Okay. In this C. elegans also, the some of the researchers are focusing on the C. elegans. It's not a tedious one. Okay, evolution of development is nematode related studies. Okay, nematode related studies are that are studied well studied on the C. elegans. Okay, in this, if you are using this elegance, we can specifically alter the genes. It is easy in the C. elegans. So only they are selecting the C. elegans for the genomic related studies having the functional counterparts in the human. Okay, so the C. elegans genes are many of the genes which are present in the C. elegans genome having the functional counterparts in the human. So which makes the extremely useful to model for the human diseases. So why they are using the C. elegans means that make a resemblance, that having the resemblance genes in the like a human. So this C. elegans are used as a human model. This model for the human diseases, not a human model, that used as a model for human diseases. So only they are taken the C. elegans. This C. elegans, elegans, that genomes having 100 million base pairs. Consists 6 pairs of chromosome, 5 pairs of autosomes with XO chromosomes in a male. Here this is the one thing, that 5 pairs of autosomes with the XO chromosomes in the male. They are having this is the important thing. This may ask in the 2-mark question. What will happen if wingless RNA is expressed in wingless expression cells from the stage when this gene is initiated its expression in a developing Drosophila embryo? Here the one I want to let you know one thing in this stage. The stage is called, here you can see, one minute I'll let you know. This lymph record related studies, and here you can, this is the embryo development stages, and this CSIR is a well studied system in the developmental biology. And the C. elegans, here the Drosophila, these are all the each and every organism group, that each and every family, they are studied some species. So this is very important thing, just study the stories. What is now I will send the developmental evolutionary studies of C. elegans and Drosophila. I will send the material separate. That is very interesting materials. Actually, last that 30 days class, that is a discussion class, that in fully portion conceptual class, in that class I was discussed the concept about the C. elegans. I was send the material of, relevant to this C. elegans. I will send that materials to you. That will help you to understand. And here the question is, what will happen if a wingless RNA this expressed in the wingless expressing gel? Okay, what is the wingless RNA? In this concept is very interesting in this latest researches, which, because it's nothing but it's complicated. In this wingless RNA, the wingless is that is one kind of pathway, that is the Wnt signaling pathway in a Drosophila is well studied. Okay, this Drosophila having the Wintless pathway represent one of the best characters of the intracellular signaling of the Drosophila. Okay, in this concept, that RNA interference, This RNAi is the RNA interference. This RNA interference screening in the Drosophila, it's a well studied one. So this is all the important thing. Wingless RNA, that is Wnt signaling pathway which involved in the intracellular signaling. This RNA interference, it's well studied in the Drosophila. Where it has happened in the Drosophila means here they are provided the answer B, that is only R, which is the they are asked the question that is expressed in the wingless expressing cells, the stages when this cell initiated its expression in the developing Drosophila embryo. The correct answer is the R. The posterior compartment of each future segment will get affected. The posterior compartment of each future segments, okay, the Drosophila having segments on this structure, this segment structures will get affected. This happened while they are doing the wingless RNA expression. Whenever doing the wingless RNAi expression expression on the Drosophila, that not able to make a segment on its own that make own compartment on their structure. This is the process. This is the output of the this wing RNA expression. This wingless RNA expression, it's helpful for the genetical screening. Genetical screening means if you make a genetical modification, means that modified wings that normal having the wings. So this is helpful for the understand the that signal transduction activations pathways, that is the wingless Wnt pathway in the Drosophila. Is all about the Drosophila. Then mutant was experimentally generated, which had wings reduced to hat tears like structures. Okay, this another important concept in the process of the ultra Bithrox gene, and also it's involved in the Hematoetic mutation. Mutant was experimentally generated. That mutant we want to generate experimentally, which had wings reduced to halter like structure. This we are all if you are a biology student, you know this halter structure. This halter the halter's wings are in it's all this flies, the flies are having the Tiftaras. Okay, Tiftaras we may know in Tamil, that is called as Tattan. Okay, that having that is wings are well structured, well interesting, that wings that give a better feel. That wings are halter's wings. In this halter wings is the flies are having that old name is it's a Tiftaras. In this Tiftara, it's flies. Its halteres are small. It's not structure modified from the king wings. Normal wings, it's a king wings. This king's wings are modified into the halteres. This why this modified into the halteres means that is there want to be change that want to face some changes in the environment. So they are pair of shaped organs. Okay, club shaped organs of the body of two orders of flies of insects provides a information about the body rotation during the flight. So this the halteres, it's are the pair of a small club shaped organ that which layer by the wing on the body of the two order of flying insect. Okay, they are present over the body, and it means that wings, it's this helpful to provide the information about the body rotation during the flying. During the flying, in this kind of the halteres, that it's first of all I was told I was told in the initial of this question, the halteres are having the modified wings. There's all the modified wings. This wings are normally in Stepsitia. Okay, Stepsitia. it's a four-winged one. This that are modified into the halteres now. Okay, Striptera, it is having a four wings that are now in the halteres. Four wings, that four wings are there. Four wings are that are modified in the halteres. And these halteres are shaped like drumsticks. This drumsticks, so it's looks. They are connected to the thorax at one end, and another end is thicker structure at the another end. These are all the connection systems of the halteres. Calteres. Okay, calteres. This overall concept about the calteres. Here they asked the mutation had wing reduced to the at some mutation. What kind of mutation make this calteres? That modification, as I am told, that is the modified one, that is modified wing. This modification happened with the ultra pitrox. This ultra pitrox is very important thing in the this calteri structure for ultra pitrox. It's kind of gene, which gene is ectopically expressed to second thoracis segment. Okay, this if you are a biology student, you all know the ultra pitrox. This is a homobox gene. This found in the all kind of insects. Okay, this gene, very important things. Ultra pitrox are it's present in the homobox gene. Very important homobox gene. This homobox gene, it's present in the all kind of insects. It's make a patterning in the morphogenes. Patterning, what is patterning? Okay, if you are taken any insect that having the head part, different head part and body part is in different segments are there. For example, if you are taken any mosquito, that mosquito also having the segments. If you are taken any insect, that insect having the loosey body, strong head, wings. This patterning of the body and morphogenes are made with this help of the ultra pitrox. This pitrox are helpful for this modification, haltery like structure formation. It's this kind of genes are very important, and here that are make this expressed in the this in the process called the thoracis segmentation. In this segmentation, the second that each and every segmentations are there, it's make segmentation, each and every insects having different segments. In that segments, it's make a change on the second segment. Okay, this make a change in the second segment, thorax segment. Okay, in this next one is called concept is the another mutation. I'm a Homeotic mutation. The another what happened for to regarding of phenotype, its answer is a D. So we want to know the hematoetic mutation. This hematoetic mutation, it's also happen in animals. Homeotic mutation. This homeotic mutation, that genes are very important, that may happen in the animal models in the many texts. Okay, this homeotic gene, it's called as the HOX gene, HOX gene. It's regulating the expression of gene. It's aiding the development and assistance of specific structures. Okay, the specific structure modification. This gene is muted. Okay, if the gene is muted, the body structure development is in a they are wrongly developed, so they are changed, that dramatically change the structure, that insect may convert into the mammal that may happen with the that Hox gene. It make a body structural change. So Homotic change. As per the statement, the qualitative like structure is a structural change. The structural change is happen with the homeotic mutation. It's a homeotic gene related one HOX. And the followings are certain segments regarding the activities of homeotic gene class A, B, C, D, C involved in the floral organ identity. Okay, very important. Very once again, I'm tell this floral in organ identity. The floral organ identity concept. Okay, yeah. They are studying the floral organ identity? This okay. In this identity gene, floral organ identity gene, why it's important? It's specifying the identification. Okay, a rose is in a rose color, jasmine is in a white color. That all those that are petals of roses numbers are high, jasmine numbers are thin. So these things are decided with the genes, particular genes that is called the homeotic genes. That having the ABC model, if you're but well studied botany student means you may know this concept ABC gene. That classes homeotic genes are that uh, function of ABC. This mutants having this triple mutants would have no gene functions, but it determine the normal floral development. As per the triple mutant, lack any organal function. If mutation is happen, it means that it may need any problems. So that are very important. Very important. Okay, here they are asking the question. Okay, what question? Which of the following combination of segment is correct? Correct is a PRS, right? Activity of A alone specifies sepals. Activity of A alone specify the sepals. Second right one is R. Activity of B and C forms stamens. Activity of the carpels. Okay, C alone makes a carpels. Okay, now this is the correct answer for this question. Now I am enter into the concept called ABC model of flower development. So in this model, a type of chromatic genes are A, B, C. Okay, here the A alone helpful for the sepals, that is the that calyx, okay, calyx, 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 sepals formation. And next concept is C alone responsible for the carpels, okay, and this carpels is the gynoeciums, gynoeciums, and C and B, C and B that are responsible for stamens, STAMENS, that is called the androeciums. A and B is responsible for petals, corolla, that is called as the corolla. So 5 points are very important. A only for sepals, C only for carpels, A and B responsible for petals, C and B responsible for stamens. These are all the 4 concepts involved in the ABC model. I think it's we can understand. Okay, next question. Okay, it's time up. Actually, I'm taking more times today. I'll continue. Okay, five. Two more questions are two. Okay, this is the last. Okay, five more questions are there. I will discuss it fast manner. For successful fertilizations in a sea urchin, as I'm told, sea urchin studies are well studied by researchers, so they may ask repeatedly. Interaction between the surface of the egg and the acrosomal protein, specifically a 30.5 kDa protein called binding, is necessary for the egg interaction. The binding is very important. It's helpful for the attachment of surface of egg and the acrosomal protein. Interaction between the surface of the egg and the acrosomal protein. The following factors could affect the interaction. What is the possible reason? P and Q. So this removal of egg jelly polysaccharide. The jelly is helpful for the attachment of egg. So if they remove the polysaccharide, that will affect the fertilization. Second is the Q, removal of binding receptor. Okay, if they are removing the binding receptor, the egg is ventillin membrane, so that is not able to bind. These two reasons are possibility uh, to affect the fertilization. Sorry, it affect the interaction and prevent the fertilization. Okay, so these reasons are prevent the fertilization. The next question is mutant embryo of Drosophila, in which one of the major sex determinant gene, sex lethal, can only undergo the default splicing, was allowed to develop. Following statements are towards explaining the determining of sex of embryo. In this part, which is the correct combination? P and S, embryo will develop into the male flay, and the sex lethal gene product directly regulates to specifically alternate splicing of double sex RNA. These are all the reason of P and R. P and S is the answer. Sorry, so S is the answer. Sexual lethal gene product regulates sex specific splicing of transformer RNA, which in the return regulated slicing of double sex RNA. These are the correct combinations. And at the two celled stage of the Canarho habits elegans. This is the C elegans. C elegans is once again this concept called C elegans. At the two cell stage of C elegans development, Blastomere was experimentally separated, allowed to proceed the development. The blastomere P1 is developed, generate all the type of cells. That C is the answer. P and R, the statements are there. That makes the initial changes. Okay, next concept is called during lens formation of the synapse. The following statements have been proposed. What is the statement proposed? R and S is right. Only the head ectoderms can respond to direct signals from the optic vessels to form a lens. Another option is the anterior neuronal plates primers that head ectoderm via PMP4 and FGF8 prior to the signals from the optic vessels development. Next final question. The Tropito is a transmembrane receptor on the fossil cells that binds to crucan protein located in the perfunctive dorsal surface of the oocyte, and it inhibits the cascade leading to nuclear localization of the dorsal protein. In the experiment, Drosophila germ lines chimeras were made by the interchanging pole cells. Germlines precursors between wild type embryo and embryos from mother homogeneous from the mutation of the Tropito gene. These transplants produce the wild type female whose eggs comes from mutant mother, trophotes deficient males, which producing eggs came from the wild mother. This the story itself having the answer. Just read again by yourself and get the answer. The answer is the 1 2 4 are the right answers, are the correct answers. 1 to 4. So whenever we are facing the CSIR, and read the question. The question itself having the answer, and just read the question and imagine yourself about the question. That question itself having the answer. So this question itself having the answer. So this is not a technical one, this is a logical one question. Torpedo, the deficient egg developed in a white type ovary produced normal embryo, and wild type eggs developed in a torpedo deficient ovary produced the ventralized embryo. Another option is trousal proteins enter into the nuclei of the drosula side of embryos, which came from the wild type eggs developed in the torpedo deficiency ovary. These are all the answers, correct answers. Finally, thank you everyone, thank you for your patience. I welcome you all to the next tomorrow day 16th class. Don't quit any classes, don't